And so today I'd like to talk about some of the work that I did as a PhD student, and also some of the work that I'm doing as a postdoc in biolanguage lab at MIT. And the title here is Engineering Biomaterials based on inspired by biology and challenges and opportunities. So this is how this talk is going to go. First of all, I like to identify some of the challenges that I think is facing the, pardon me? Oh, sorry. Go ahead. Well, identify some of the challenges that's facing this field of biomaterials engineering. And then I'll describe two approaches that we think might be promising to tackle these challenges. And I'll give specific examples for each of those two approaches. And then in the end, I'll spend some time talking about what the future I think will be like in this field, and also my own research. So over the last number of years, biomaterials have really experienced a lot of tremendous successes in terms of making functional devices and use them to change people's lives. So here I just like to give a few examples. So here you see a total artificial hip. So it's made of a variety of different materials, including such as metals, metal alloys, ceramics, and polymers, engineered in such a way that it supports the skeleton and it functions like a joint. And here is an example of a total artificial heart, which is really an engineering marvel. It's made of, again, a number of different types of materials, including metals and polymers. And it's engineered in such a way so that it can perform the very important function of a heart, that is to pump blood. And here you see a different kinds of material, which is based on polymers, and it's a soft material. It absorbs a lot of water, and you can see that it's very flexible, very soft. And this is actually the soft contact lens. It's made of the material is called a hydrogel because it absorbs a lot of water. So the function of this material is to correct vision. And here another example is polymers containing drugs and in the matrix, and slowly release them to modulate disease processes. So far, the success of all of these devices have really centered on two issues: the so-called biocompatibility and performance. And these two are interrelated. So biocompatibility nowadays refers to that material, once it's inside the body, it doesn't do harm to your body. It doesn't create any inflammation, doesn't create immune response. And performance, of course, these different devices have their own desired being a replacement or enhancement of physiological functions. But again, where do we go from here? So again, I think there are tremendous drawbacks as well, even with these successful examples. Again, in those two aspects, biocompatibility and performance. For example, this total hip here, it's only good for about 10, maybe 15 years. And there could be a lot of problems associated with it. You can get loosening, you can get degeneration of bones because you're essentially shedding a lot of stress. And it can be there can be very slow chronic inflammation, and in this case, artificial heart, it's in contact with blood and it's pumping blood in such a high flow rate. Inevitably there's going to be damage of the blood components. So these are the challenges I think that is really facing the biomaterials community. These are the two things I identify, of course. First of all, how do we make biomaterials with well-defined structures and functions? Right now, we're not really very good in doing that. So for example, if you're making a polymer, and you're making a polymer using the traditional chemical synthesis, and this is what you'll get in terms of the size of the product you're getting. I call this a molecular heterogeneity. which essentially is a Gaussian distribution of a different chain length. And the species here involved can be a huge number, like millions or hundreds of thousands. So you think you're making one polymer, like polyethylene, but actually you're really dealing with many, many different kinds of polymers, each of them with a very distinct structure and property. Of course, that's going to influence any biological applications. And on another different scale, on a microscopic scale, also there's tremendous heterogeneity. What you see here is an example of polylactic acid. It's one of the FDA-approved material, and in this case, it's a tissue engineering scaffold, and you have liver cells sitting on those. And you see those fibers, they really go all kinds of directions, and there's very little control over that. The space between the fibers is very heterogeneous. So this, of course, is going to have a tremendous influence on the performance of this material as a tissue engineering scaffold. So, and the second challenge I see is really that we're making biomaterials. We're not just making any materials. So how do we make those biomaterials that can be sort of interactive with the biological environment, not just really sitting there passively? So I see this, we need really an update of the definition of biocompatibility from the current point of view, seeing an implant as a passive, inert, non-toxic, non-immunogenic material, really to upgrade to something that's active and really can be smart in some ways to communicate with the biological environment and modulate the biological environment. So, all right, so these are the two approaches that we are using to deal with those two challenges. So the first approach really is based on understanding the biological molecular biomolecules and how they are put together and how they self-assemble and how they interact with each other to form these architectures. So like from here is an example. You start from biomolecules, you have the DNA, which encodes all the genetic information, and then you have a protein, and the proteins will self-assemble to form the super molecular structure. In this case, it's an actin filament. And these superstructures further form a hierarchic structure, in this case, like a cytoskeleton. So by contemplating on the properties and structures of these biological molecules and architectures, we decided that we were actually going to use these biomolecules, such as proteins, as building blocks to make materials. And the way we prepare these materials is using the well-developed recombinant DNA technology. So by this approach, ideally, or in principle, one should be able to produce a material with exactly defined size and composition and structure. This is in large contrast to the more traditional polymer technology. But of course, the traditional polymer chemistry has a long tradition. A lot of the basic questions has been answered. So we're going to actually use a combination of both of these two to combine these two technologies to make materials. And the driving force of the materials can be either chemistry or more of biological-oriented processes, such as self-assembly. And the second approach is really based on the understanding of biological processes and systems. So in this case, we start from a clinically relevant problem. For example, let's say you're treating cancer, or you want to engineer a tissue. So you go back and understand really on the molecular basis, cellular basis, or tissue organ basis, and ultimately the systemic systems basis of all these processes involved in those diseases that you want to treat, and then go back to see what elements you need to be put into the material so that you can achieve your desired therapeutic goal. So now I'm going to move on to give some examples. So the first approach, I will give two examples. One is about using engineered protein domains to drive microscopic structural change of polymer hydrogels. The other example I'll go through very briefly is to use biological templating and self-assembly processes to form inorganic nanomaterials. For the second approach, I will use an example using biopolymer, degradable polymer microspheres for the controlled delivery of DNA vaccines. So for the first example, here's some background about hydrogels. So hydrogels are really polymer chains that are tied together through these so-called cross-links. And these are all very hydrophilic chains. They absorb a lot of water. So instead of dissolving in buffer, they would swell and maintain a certain shape. So in the end, there's an equilibrium state where you achieve a thermodynamic equilibrium between the hydration of the polymer and the surrounding solvent. And this type of material has already been used extensively as in the biomedical devices, all kinds of applications shown here. And a very interesting and intrinsic property of all hydrogels is called phase transition. So this is similar to really the physical phase transitions that we see, for example, water freezing or water evaporation. But it's done at a different scale with a different identity. So here, for example, here you have a hydrogel which is in equilibrium with a solvent, and then by changing the environment, such as these different factors, in this case, you change how the polymer chain interacts with the solvent, and you can actually drive the solvent away from the polymer. You get sort of a localized precipitation, and that's called a phase transition of a hydrogel. And usually it's accompanied by a change in volume, a change in physical properties such as optical properties. And these phase transition process is very specific to a specific stimuli. For example, something that's responsive, something that occurs at a certain temperature would not occur at a different temperature. It's very specific. And the process is also highly cooperative and, in many cases, reversible. So the currently available phase transition-based polymer materials really doesn't leave too much room for engineering. So we came up with a new approach where we call this hybrid system, where we have essentially the system will contain two different components. One is based on synthetic materials. So we're intent to use this as a mechanical support and to absorb water to keep everything swelling in water and to minimize non-specific interactions involved in the second component, which is based on proteins. And the proteins here is actually the vital component where we're trying to take advantage of the stimuli responsive behaviors of protein conformational change to drive a macroscopic property change of these hydrogels. So we started with a relatively simple protein folding motif called the Coil Coil. So what is a Coil Coil? So here is a whole bunch of native proteins. They have very diverse functions and structures, but all of them contain part of it, this Coil Coil motif. So it's basically two or more orthohelices winding around each other to form a superhelix. And the beauty of this motif is that a lot of the biology has been done so that we know a great deal about how the amino acid sequence correlates with the structure of this motif. So for example, here in this case, if you're looking down from two of the helices, and you will see the first and the four positions of the amino acids are often hydrophobic residues, so they sort of serve as a zipper to glue these two helices together to make it stable. So then we moved on to produced two coil proteins we call 1 and 2, and we made these using the recombinant technology. We constructed the recombinant genes and put them in E. coli and expressed them and purified them from bacterial culture. And then we looked at the secondary structure of these proteins, so we verified that they do fold up very correctly into alpha helices. And we found that one of the proteins actually unfolds, has a thermal temperature-related conformational change at about 37 degrees, whereas the other one doesn't really have any change. So then we moved on to construct this material, this hydrogel, three-dimensional material, where we have a synthetic polymer backbone that is able to complex with a metal ion. And then we engineered histidine residues on the ends of these proteins so that they can bind to these ions and form this cross-link. So interestingly, when we increase the temperature, we saw that in the case of the hydrogel, which contains the first protein, which is capable of undergoing conformational change, we saw this dramatic collapse of gels from a fully swollen state to almost 10% of its original volume. And at the same time, we observed also a dramatic change in optical properties. So actually, the other gel, which contains the other protein, really didn't have any change. So what's happening here? So this is what we think, this is our hypothesis. So we start with the really long extended alpha helices, rod-like structures. And if you unfold these things, and they form these random structures. And these random structures occupy less amount of volume, and that's the driving force which brings the hydrogel to a collapse. And to illustrate a little bit better here, so let's say if you have the same number of amino acids in a protein, and depending on the different conformations, they occupy different amount of space. And if you move from one conformation to a different conformation, that change in volume would bring about the change in the hydrogels. So then we moved on to construct some more complex structures. In this case, these are block proteins. So we're taking a protein segment of a native protein called kinesin, which is one of the motor proteins in charge of intracellular transport, and we cloned that. This part of the protein sequence consists of part of a helix and part of a random coil. And we made one copy of the gene, and then we also duplicated. We put two copies side by side with each other, and also then we moved on to make three repeats as well. And we were successful to be able to express these proteins in bacteria and purify them. So we did a number of characterizations on these proteins, looking at secondary structures and how they associate with each other in solution, and how much space they take up in a water-based solution. And we came up with this model here. So remember this, we call this KS1, which contains one copy of that gene that we taken from kinesin, and compared that with 2 and 3 copies. And really interesting here is that we think that these, rather than forming a linear structure, they actually fold up within each other to form these more compact structures. So, and also very interestingly, they display sort of this hierarchy structure, whereas this trimer can be viewed as a combination of a dimer, which is shown here, plus a monomer sitting on top of it. And indeed, the behavior or the thermal transition behavior of this trimer is the sum of the monomer and the dimer. And so we used this model, although it's hypothetical, but we were able to explain how the proteins behave in the context of a three-dimensional hydrogel. So this is really good. And then I'd like to move on to a different kind of material, which is sensitive to calcium. What you're looking at here is a very primitive single-cell organism called Vorticella. So this is a cell body, and it lives underneath water, and it fastens its cell body through this long organelle. It's called spasming to a solid support, such as a leaf. And then in the presence of trace amount of calcium ion, it contracts dramatically. And if you take away the calcium, it relaxes back. And the interesting thing about this motility is that it's not dependent on ATP, unlike, for example, muscle contraction. And basically, this organelle consists of aligned fibers, protein-based fibers, and a hydrophilic sheath wrapped around it. So the mechanism of this contraction is really not known right now, but the working hypothesis is that you have these well-aligned fibers, and in the presence of calcium, it somehow disorganizes the well-alignment fibers and causing the fiber to contract. So here I'd like to show you a movie of the contraction. This is real time, and the contraction process is completed in 1/60th of a second. It's one of the most violent biological motility in biology. And the relaxation is kind of a little bit slower. It takes about a couple seconds. All right. So later on, only in the recent years, people found isolated and also later cloned a gene which encodes a protein they called spasmin. And really, the spasmin is found throughout these organelles. It's all over the place. And it has a very strong sequence homology with a well-known calcium-binding protein, calmodulin. And the way calmodulin works is that it's a dumbbell shape, and it binds with 4 calcium ions. And in the presence of a target peptide, which is sort of perpendicular to the slide, it wraps around it to form this a very compact structure. This is also one of the most, or the largest protein conformational changes which has ever been observed in biology. So the question now remains is, does spasmin work the similar ways, similarly to as calmodulin, or does it have a distinct mechanism? And more relevant to us is that we want to develop materials. So what kind of engineering principles can we learn from these structures so we can make better materials? So we produced a recombinant spasmin in the lab using recombinant techniques, and also constructed fusion proteins of GST with these spasmin. And we compared spasmin fusion protein with calmodulin fusion protein using a technology, surface-based technology called quartz crystal microbalance. Basically, you have a quartz crystal, and you coat the crystal with a layer of proteins. And if you have any protein structural change, it's going to change the properties of the crystal, and it's very sensitive. So the bottom line is that we found that this spasmin behaves very, very differently than calmodulin. It has a very dramatic conformational change in the presence of 1 millimolar of calcium ion. So this is really very interesting. And then, just by chance, we sort of observed that when we start with the GST spasmin fusion protein and we add calcium to it, and if we shine UV light to it for just a few minutes, we observe these formation of these type of structures. These are very extensive, large ribbon-like structures. And some of them are really large in terms of size, and some of them are really thin. But these are highly regular structures. There are nodules sort of along these fibers. And the formation of these structures is highly dependent on the presence of calcium. If you treat this with EDTA, you remove the calcium, this is what you end up. You have all these globules left. You don't have these fiber structures anymore. And the dimension of these fibers is actually roughly on the same scale as the spasmogene found in the Vorticella. So we think that so far this is all has been really interesting. We have a working hypothesis in terms of how this works based on these data. So we think that this protein, very different from calmodulin, it actually uses the calcium binding to change its conformational change and use that to induce motility. So this is also some work ongoing that we're using these principles and engineering variations of these proteins to make calcium-sensitive materials. So now I'm done with the first example here to illustrate this approach, and then I'll go to this more nanotechnology-related approach. This we feel that the bioinspired engineering approach is not only applicable to biomaterials and biological molecules, but also it'll be interesting to take advantage of these methodologies to engineer inorganic nanomaterials. So these are just a number of examples of different nanostructures that people have been able to make so far. And almost a lot of these structures are made using the traditional chemical synthesis, where we're often involving high temperature, high pressure, the use of catalysts, which is really not very compatible with biomolecules, bioprocesses. So we decided to take an approach that's based on protein assembly and templating to make these materials. So we decided that we're going to focus on a very common protein called actin, which is a very important cytoskeletal protein. And this is a monomer called G-actin. And in the presence of physiological salt, it self-assembles, binds with ATP, it self-assembles into these extensive fibers shown here. And so we have developed ways to to use this process, and this process is very reversible. If you take away the salt, it goes back to the monomer. So we've been able to construct metal clusters along these fibers, and in this case, it's really a few micrometers long, which is really long, and in some cases, the arrangement of these metal clusters are really straight. So, and we're trying to improve on the methods of these to hopefully we can engineer something like a wire that's really capable of conducting current. So, of course, you can think about using these materials as microelectronics, but also we're more interested in is to a combination of these nanostructures with bioentities such as biomolecules or even cells. All right, so now I'm going to move on to the second approach. Whoops, let's see. All right, talking about DNA vaccines. So what is a DNA vaccine? The traditional vaccine is based on attenuated viruses, pathogen antigens such as proteins and peptides. And DNA vaccine really starts with a piece of DNA. And the DNA encodes the antigen that you want to immunize against. And you put it into a plasmid and you replicate that, proliferate that, and then you inject the plasmid into an animal. And hopefully the gene will get expressed, the protein will be produced and presented so that you will get an immune response. So in terms of the immune system, how the immune system works inside the cells, this is just two typical classic two pathways. One is really involving that on this half of the graph here is the so-called exogenous pathway, where this is more of how the traditional vaccination works. So you have an antigen that's coming from outside of the body and being taken up by cells and being processed, chopped up, loaded onto these molecules called MHC class 2 molecules, and then comes back to the cell membrane, which elicits an immune response. And so DNA vaccine works in a different way. Actually, it takes advantage of the other pathway, which is where you have the antigen, the protein, produced inside the cells, and that gets processed and chopped up and loaded onto the MHC class 1 molecules, and then goes back to the membrane and create an immune response. So it is known that class 2 pathway is really important in eliciting antibody-related immune response, whereas you will really need class 1 pathway to elicit both the antibody-mediated as well as cell-based cellular response. So this is how the whole immune system works. You have these two different humoral response and cellular response. But really the important part of these two pathways is how the antigen is being made and presented. So this is where we think that we should be able to contribute to, is that our hypothesis is, after contemplating on these immunological processes, the hypothesis is that if we can achieve a way to selectively target certain cells inside the body that are professional cells that are made to present and process antigens, then we should be able to get a better immune response. So the way we're trying to do this is by using polymer microspheres encapsulating these DNA vaccines and make these microspheres into a certain size, for example, 1 to 10 microns in diameter, so that they're too big for normal cells to be eaten up, but they're perfect for example, dendritic cells or macrophages to be taken up and processed. So let's imagine if these microspheres are being internalized by this cell, these DNA will can somehow escape these vesicles and then eventually reach to the nucleus and gets expressed and processed, displayed on the cell membrane, and then the immune response can be elicited. So the polymers that we have been using is really based on this new polymer called polyethyl and we are trying to and we're using this other polymer, PLGA, polylactic glycolic acid, as a comparison. So this polymer here is probably the most often used biodegradable polymer so far, because first of all, it's very biocompatible and it's approved by the FDA for in vivo use. But the problem with this polymer is that you make an implant out of it, the degradation of this polymer really occurs throughout the entire matrix. So, and in that process, you generate a very acidic degradation product, which has the great potential of damaging the DNA inside these particles. So whereas this new polymer that we've been working on in collaboration with other people is that this really doesn't degrade in this case it's a bulk erosion. In this case, the polymer really resembles a bar of soap, whereas only the surface, which is in contact with the solvent, gets degraded and falls apart, and whereas the interiors of the polymer microsphere remains intact. And by doing so, there will not be any acidic degradation product accumulation, so that the DNA conformation will be maintained throughout the entire process. And also by manipulating the chemistry of this polymer, one can incorporate different triggering molecules so that you can sort of modulate these processes, the degradation, by changing the environment. So these are the number of different monomers that we have incorporated into these polymers. So for the first two categories are really the basics of forming the bond, the polymer backbone. And these different variations of the second polymer gives you very good flexibility in terms of controlling the hydrophilicity and rigidity of the polymer chains. And this polymer is what we call a latent acid polymer. So once during degradation, this polymer is generated, and it's an acid. So and then that serves as a catalyst. So you're speeding up the polymer degradation as the degradation process goes along. So it's a self-accelerated degradation. And we decided to incorporate another monomer which contains a tertiary amino group, which is positive charge at neutral pH. So what we hope is that because DNAs are large molecules that are negatively charged, so by incorporating this positive charge, there will create some interactions between the polymer and the DNA. So I'd like to have you to focus on two polymers that we made. One has all these components except this last amine monomer, and P2 has a certain fraction of the amine monomer. So we encapsulated DNA into polymer microspheres by using a very well-defined double emulsion technique. And this is just to show that we're able to make these nice spherical particles with very smooth surfaces, and we can have very good control over the size of these particles. In this case, it's centered, the average size is about 5 microns, and the distribution is fairly narrow. Okay, so then we looked at how the DNA behaves inside the particles, and we extracted the DNA out of the particles at different time points to see if these DNAs remain active in terms of the conformation. We run gels to see if they maintain the active conformation, because that's really important in terms of protein expression. So the bottom line is that we saw that a large portion of the DNA remains as the most active form, and even up to 3-5 days of release. And we quantified the amount of DNA released in terms of with time, and we studied different pH buffers and how different pH affects the release kinetics. And we found this very interesting phenomenon that both of these two polymers release very slowly, very little amount of DNA at neutral pH within, of course, a certain period of time. But if you change the pH to slightly acidic, for P1, there's a dramatic, very fast release burst, and it releases all of its DNA. Whereas P2 has a considerable lag time, and then it starts to take off. So this difference here has to do with the presence of those amine-containing polymers in P2 polymer. And the reason why we're interested in these two different pHs, because remember one of the figures I showed, the hypothesis where you have the spheres go throughout the cells, and they first go into the cell, they go into these vesicles called phagosomes that are kind of slightly acidic. So we're trying to mimic really the biological processes where the microspheres, when they're inside the cells, what kind of environment that they're going to be in, and to try to simulate that in vitro. And then we looked at how these polymer microspheres degrade inside cells. This is a macrophage cell line. After incubation with polymers for 4 and 7 days, this is using fluorescent confocal microscopy. So the conclusion is that we see with time, we see progressive degradation of both of these two polymers. And P1 actually degrades slightly faster than P2, which is in agreement to what we observed in vitro. And currently, we're doing animal testing with using model antigens, looking at the production of antibodies and also the ability to elicit cellular responses. Basically, we're looking at the ability of activating T cells using, again, a model antigen. And the preliminary data showed that compared with control experiments where you don't, in mice, that there was no treatment, or if you treat it, immunize it with DNA, naked DNA without any polymers, they don't really have any activation. So using these P1, P2, and PLGA polymers, we got fairly good activation, but both of the P1 and P2 do considerably better than PLGA by itself. So this is really encouraging, and this work is ongoing. We're also going to be doing, actually in the process of doing a tumor challenge experiment where you immunize the mice first and then you inoculate them with tumor cells and see if we can really prevent tumor from growing. All right, now I'd like to thank a number of people here at MIT. My advisor, Bob Langer, has been really extremely supportive and providing a wonderful environment for people in the lab to pursue their sort of their own interest. And then a number of wonderful collaborators at MIT. This is in the Langer lab and also some other neighboring labs in biology and also in the Whitehead Institute that helped us on various aspects of these projects. And also my advisor at the University of Utah and a number of people, faculty members there, which helped me throughout the years when I was a student there. So, and these different people contributed and collaborated with on different aspects of the project and acknowledge the support for my postdoc study by the NIH. Now I'd like to move on to talk a little bit about what I see the future of this field is and how my research is going to contribute to. So this slide pretty much summarizes the approach that I'm going to be taking. So really the important point here is that this is a bio-inspired approach. So again, really understanding how biology is made up on the molecular level and hierarchy level, interactions between different biological components, and the processes involved in biology. And this is one starting point. Another starting point is based on applications. So really understand what the problems are that have very potential to have clinically important impact, and then go back to design rationally these materials to modulate these processes. And the approach in terms of engineering really is a unique combination of biosynthesis and the polymer chemistry. So this is what I see as really unique in this approach, because by incorporating biology into the more traditional polymer materials, it really opens up a lot of opportunities for making interactive materials, as I talked about in the beginning. So these are, just go real briefly about the three different things that I thought about that I may be interested in pursuing. The first one is really based on design smart materials based on biomolecular structures and interactions. So if you look at these three different things, proteins, cytoskeletons, and the extracellular matrix, you will find that the common feature of these three very different identities on different scales have really very common features, is that they all are hydrogel structures. So these are the characteristic properties of a typical hydrogel, and you can find all of these properties in these three different materials. Have the proteins, have these cytoskeletons, skeletons, and then you have these cells sitting in the middle of a three-dimensional extracellular matrix. So after looking at these, I'm thinking it'd be really interesting to make materials based on these properties and to really mimic the structure and function of these identities. So expanding on some of the earlier work that we did, really to expand these to incorporate a large different varieties of biomolecular structures and interactions. So for example, in this case, if you create a polymer hydrogel based on a protein conformational change, if you change the conformation, you change the property of the gel. And in this particular type, if you start with an aggregated protein and somehow trigger, put in the trigger and dissociate these proteins, you will get a property change in the gel. And in this case, if you have a protein and a ligand, and if you disrupt the ligand in certain ways, you will change the property of the gel. This is also a similar case in this, but more complex. And I have some examples where there are certain, actually there are many, many different examples in biology that one can take advantage of creating these materials. So these are some of the long-term things that could be applicable by this technology, such as tissue engineering scaffold, drug delivery devices, and actually incorporate these structures in microfluidic devices is going to be also very interesting. Because of the presence of biomolecules and the conformational change, one can envision that these could be useful as biosensors. And these are more practical applications. And from the more basic science point of view, first of all, it's just be interesting to make new materials. And secondly, I believe that this could be a really useful model system to look at how proteins on the molecular basis, they would change their structure and how to respond to stress on a nanometer scale. And also, more importantly, more relevant to tissue engineering is the process of mechanotransduction. This could be also another model system to study that. All right. So now I'll move on to the second proposed topic is for tissue engineering or regenerative medicine. So as all of you know that there are several key components in tissue engineering in generating a tissue or organ. So you have the cell source, which is interesting. You can use primary cells or stem cells or other pluripotent cells. And there have to be soluble factors such as growth factors. You have to give the different kinds of growth factors to cells at different time course and different patterns, different doses, and very complex. And then another component is mechanical stimuli, where you have different kinds of patterns and different shear and different stress. So, and this is where I stand, the polymer scaffold. And what I hope to do is to engineer polymer scaffolds so that they're able to integrate all these components to achieve this final goal of engineering a tissue. So this is a hypothesis, what I call a designer three-dimensional scaffold, whereby really using the protein engineering technology, as I described before, one is able to, should be able to incorporate diverse different functionalities into a single scaffold. For example, in this case, you can envision that for mechanical properties, you should be able to incorporate different modules. Some of them are soft, some of them are hard, and in combination of these two different things, you would get desirable mechanical properties of the scaffold. And of course, you can think about if you want to interact with cells, you can put different kinds of cell adhesion modules into the chain. And also, you want to be able to have the scaffold to be able to be remodeled. So to put in enzymatically degradation sites is going to be important. And also, it should be also possible to incorporate growth factors, for example, like in this case is a tethered growth factor as part of the scaffold rather than adding a soluble growth factor, which is going to be really easily diffused out and being consumed. So, and if necessary, a synthetic polymer can be incorporated just to maintain, again, the mechanical strength and adding in water solubility, swallowability of the scaffold through these very highly well-defined crossings. So these are certain things that I'll be looking at to characterize these scaffolds and eventually to move, hopefully to move from the in vivo to in vitro to in vivo. And I would like to really to build a platform technology so that depending on different target tissues one wants to regenerate, to really go back and tailor different compositions of these materials so that they're individually adapted for a particular tissue. All right, so the last last field would be in gene therapy. So this entire field of gene therapy is really bothered by one issue, that is the delivery problem. That's really the bottleneck of the field. So you can imagine that if you want to get your gene, your therapeutic gene to where you want, before you even reach the cells, you have to deal with all these different problems. This is more like system-related problems in terms of stability and biodistribution, where you have the genes located. And let's say even if you can achieve all these goals and you can sort of target your DNA, your gene to a particular cell, and they can go into the cells, there are also numerous barriers and processes these genes have to go through so that they can be successfully expressed. So how do you overcome all these different issues and all these different hurdles at different scales? So that's the challenge right now. So again, a summary of the state of the art of the gene delivery vectors. A lot of the viruses have been used for vectors. Of course, they're extremely efficient, and in many times they can be permanently integrated into the genome, and the expression level is very high. But a very serious drawback is the safety issue. Many of these viruses really can create problems. So on the other hand, other non-viral vectors, such as based on these molecules, lipids, polymers, peptides, and proteins, they're in general very safe to use, but they're also very low in efficiency, and the gene expression is often very transient. So, and also their structure is really not very well defined, especially compared to a virus. And these materials are really very heterogeneous, like I described in the beginning of the talk. And it's also very challenging to incorporate functionalities to these systems. So again, learning from biology, here you see a virus, and this is how a virus looks like. And these are proteins that are on the viral membranes, and that are in charge of either targeting or help the virus get inside the cells and get trafficking, move inside the cells and get the gene expressed. So, and here's another example of a naturally occurring protein family that are built to bind with DNA. These are transcription factors. You have these modular type of organization. You have a DNA binding domain. You have another functional domain connected by a linker. So our hypothesis is, what if we can make a structurally mimic of these materials? And hopefully we can also get the functions as well. So here's the hypothesis. I imagine that one can make a modular molecule based on different functionalities. For example, you can have a domain that can bind to DNA, and you can have also other domains that can be in charge of targeting to specific cells or mediating trafficking within the cells. And you have these linkers to attach them, and through self-assembly, you would end up with this nanometer-sized complex, so that all these functionalities are exposed, and the DNA is really protected in the interior of the complex. So this sort of mimics a real virus structure, except it's made of synthetic materials. And there are a few things that one should consider, and eventually I hope that it'll be really nice to move this to the clinic so that it can be really useful. And of course, delivering of DNA has very tremendous medical applications, and hopefully this will have a very clinically relevant impact. So that's the end of the talk, and thanks very much. I'm ready to take any questions if you have. Thank you.
Are there any questions? So you did the calcium sensitive
Yes.
change in conformation reverse. Can it go in both directions?
In terms of the organism,
Yeah.
yes, it is. It's very reversible.
What
Yes.
about in your synthetic?
in our synthetic system, we haven't really looked at directly the calcium sensitivity of the material. What we so far observed was that the formation or the self-assembly of that extensive material is very much dependent on the presence of calcium. And that formation of the material is reversible, sort of. You have formation and you take away the calcium, it doesn't exist anymore. So the next step that we're actually doing is, can we make a material that really mimics that contraction process that we observed in the real biological organism? So that's work ongoing.
What about
yep.
the temperature sensitive, the unzipping of the coils?
Right.
Is that reversible too?
That is not reversible in those two examples I presented to you. And there are reasons for this. I think, first of all, protein, if you unfold a protein, there's a large chance that it may not fold back. It's just a kinetic factor that's probably limiting it. Secondly, and we're not just looking at a protein that's setting in solution by itself. You have all these hydrophilic polymers around it, and that the presence of these polymer chains will restrict the three-dimensional flexibility of these protein structures, so they may not be able to rotate and fold back to its proper state. And lastly, because of the unfolding process, some of the hydrophobic residues on the protein are exposed, and they can interact with each other non-specifically, so that they would actually form something really compact, more like a sphere structures, and they may even come out of the solution. So that process, of course, is very hard to be reversible. And in terms of applications, in certain applications, it would be nice to have a reversible process, but in certain applications, if you just want to have a one-shot type of use, then it may necessarily have to be the case.
My question has to do with this repeat that you chuck out from the kinesin tail, and you have presented hypothetical model, increasing number of repeats, and it forms certain structure.
That's right.
And you say it's hypothetical and allow you to, I guess, explain certain experimental result.
That's right.
Of the hydrogel.
Right.
How would you go about testing that the structure indeed is what you propose?
That's right. Of course, ultimately, if one can do crystallography and really solve the structures, that's going to basically answer your question. But we thought about this, and it's just sort of out of our range, and we're really our really main goal is to engineer the material. So,
pose the question in a different way. Is this hypothetical structure the only model that could explain your data, or is there an alternative model that could explain as well the data?
Well, based on the data we have, we think this is the most reasonable model. There might be some other models, but I think, as far as we're concerned, this is the most plausible model. Yeah. Yeah, go ahead.
How do you expect the lifetime of these smart materials with proteins and peptides would be like? Because proteins and peptides may be susceptible to oxidation and reduction in normal environment.
So the question is how stable are these proteins are? Okay. Of course, that depends on what kind of protein you have. Some of the proteins are extremely stable, and some of them are not. So it depends on what type of application you want to use. For example, in terms of tissue engineering scaffold, eventually you want the scaffold to go away. So then the protein, you don't want it to be there forever. So you want to go away, to be degraded, and actually, you may want to deliberately put into some elements so that the degradation can be controlled and triggered. In terms of other applications, again, depending on the exact configuration of the material, the stability issue is, as far as I see it, is not too much of a problem.
One last question.
Yeah.
As you point out, these hybrid environmentally responsive materials are very promising. There's still two major challenges that need to be addressed. I want to see what your ideas are. The first one is that for many applications, it's extremely difficult to actually control the stimulus that triggers the response. So if you're looking at the calcium, or if you're looking at some of these temperature systems, it's different to control it in vivo for a real device application. And the other issue is that many of these hybrid materials, by the nature of using proteins, could elicit an immune response, particularly since many of the proteins and sequences are intracellular components that the body has not been exposed to.
Right, thanks. The first question is, so let's say if you want to use the calcium responsive material in vivo, of course you have a lot of calcium ions present inside the cells. Is that going to be a problem? So my answer is, in that particular case, it's most probably not going to be used in vivo. I see that as an opportunity rather than a challenge, is that this approach really allows you to incorporate, by using different proteins, so that you can have really biological specificity, which synthetic materials is really not capable of doing. So for example, if you want to really use something inside the body, and you've got to really understand what is special about where you want to put your implant or material, so that the response will be only specific to that particular part of the body or any particular cell type. For example, if you really want to do something with tumor, for example, targeting a drug to a tumor, you want to have the targeting mechanism to be very specific to that particular tumor. So second question is the potential immunogenicity of these proteins. First of all, depending on the application, if you are not going to eventually implant these materials inside the body, that's not an issue. Secondly, if you do incorporate these inside the body, there can be a number of different approaches to do. First of all, you can use humanized proteins. You can use proteins directly, part of the human protein. And then, of course, if we settle on a particular application, we're ready to really mass produce that material. And the very first thing we should be doing is really to test that protein with an animal model, see if you have any immunogenicity there. So that's a very good question.
Okay, well, thank you very much.
All right, thanks.